Deep inside the human skull sits a structure called the petrous, the densest bone in the body. Geneticists discovered it preserves DNA with a fidelity nothing else can match. What it revealed about the Cherokee changed one of the oldest ideas in American archaeology. For a long time, the archaeological picture of the Cherokee made complete sense. Town sites scattered across the Blue Ridge, pottery traditions rooted in the regional sequence, agricultural patterns shared with neighboring nations, trade goods moving along the same river networks that connected peoples from the Gulf Coast to the Piedmont. Everything material about Cherokee life fit the southeastern world so naturally that the question of origins rarely seemed urgent. The assumption was continuity. A people so deeply woven into a landscape must have grown from it. Then there was the language. Cherokee belongs to the Iroquoian family. That same family produced Mohawk, Seneca, Cayuga, Onondaga, and Oneida, the languages of the Haudenosaunee Confederacy, whose heartland ran from the Mohawk Valley to the shores of Lake Erie. No other Iroquoian language has ever been found anywhere near the southern Appalachians. The gap between the Cherokee homeland and the nearest Iroquoian-speaking community was more than 800 miles. That is not linguistic drift. That is a displacement. Linguists had been aware of the problem since the 19th century. By comparing vocabulary, grammar, and sound patterns across the Iroquoian family, researchers estimated that Cherokee had been separated from its northern relatives for roughly 3,500 to 4,000 years. The split fell somewhere in the late Archaic period, well before the Mississippian chiefdoms, well before any of the cultures the historical record names. Something had moved. The question was what, and the tools available to answer it, pottery distributions, settlement patterns, stone tool typologies, could not reach far enough into that silence. Then mitochondrial DNA research reached North America in force. Mitochondrial DNA passes from mother to child, accumulating mutations at a measurable rate. That allows researchers to group populations into lineages called haplogroups and estimate when those lineages diverged. By the late 1980s and through the 90s, research teams had cataloged maternal lineages across Native American populations from the Arctic to Patagonia. 4 founding haplogroups emerged from that work, labeled A, B, C, and D. Each had clear counterparts in East Asian populations, each consistent with a migration across the Beringia land bridge from Siberia during the last ice age. The model was coherent, well-supported, and broadly accepted. It accounted for 97% of Native American maternal lineages. The remaining 3% refused to fit. A 5th lineage appeared repeatedly across different populations. Haplogroup X. Unlike the other 4, it had no obvious parallel in Siberia or East Asia. Its closest relatives lived in Europe, the Caucasus, and the ancient Near East. That mismatch provoked serious misuse almost immediately. Certain groups seized on the European distribution to argue that Native Americans descended from ancient Mediterranean or Middle Eastern colonizers, a claim used to cast doubt on indigenous identity and land rights. The evidence does not support that argument, not even remotely. In 1999, 2 independent research teams closed the door on that interpretation. Michael Brown and colleagues at Emory University and David Glenn Smith's group at the University of California, Davis, had each spent years mapping haplogroup X across Native American and Eurasian populations. Both arrived at the same result. The Native American variant, designated X2A, had diverged from its Eurasian relatives at least 15,000 years ago, before the first Americans walked south from Beringia. It crossed with the founding populations themselves, and then went its own way, accumulating mutations that make it genetically distinct from any European or Near Eastern lineage alive today. The colonization narrative is not supported by the data. It never has been. What mattered for the Cherokee was where haplogroup X appeared once the continental distribution was mapped. Smith's team found that X2A was not spread evenly across North America. The lineage clustered. Frequencies were highest in the Great Lakes Basin, concentrated among Algonquian and Iroquoian-speaking peoples. At lower but consistent levels, it also appeared in Cherokee populations in the southern Appalachians. The geographic shape of that distribution was not random. It traced the same territory that linguists had been pointing at for over a century: the Great Lakes Corridor, the northeastern woodlands, the region where Proto-Iroquoian speakers are estimated to have lived before the Cherokee branch separated. Two independent lines of inquiry, one built from grammatical reconstruction and sound change rates, the other from mutations cataloged in mitochondrial sequences, were converging on the same coordinates. If that convergence seems significant, what came next made it harder to dismiss. A few years after those haplogroup X studies, Ripon Malhi at the University of California, Davis assembled the most comprehensive continental inventory of Native American mitochondrial DNA ever attempted. 1,612 samples drawn from dozens of populations spanning the full length of North America. 395 fully sequenced hypervariable segments, each representing a thread of maternal descent stretching back thousands of years. When Malhi's team applied spatial autocorrelation analysis, a method that tests whether a genetic marker is more geographically concentrated than chance alone would produce, the continent organized itself into three distinct zones. Haplogroup A had expanded across the Arctic. Haplogroup B clustered in the southwest. Haplogroup X had expanded around the Great Lakes. Place that map beside the documented distribution of Iroquoian languages before European contact. The overlap holds. Then add a third layer: Cherokee oral tradition. The Kituwa Foundation narratives, carried across generations and recorded by outside observers who generally dismissed them as legend, describe a northern homeland and a southward journey into the mountains. Genetics, linguistics, and indigenous oral history had each independently preserved the memory of the same geographic displacement. If tracing evidence like this across disciplines is the kind of investigation that pulls you in, this is the channel for it. In 2011, the picture deepened from an unexpected direction. Deborah Lawrence and Brian Kemp at Washington State University were analyzing skeletal material from 3 Arikara village sites along the Missouri River in South Dakota, occupied from 1600 to 1832 of the Common Era. The Arikara speak Caddoan, a language family with no established relationship to Iroquoian or Siouan. There was no theoretical basis to anticipate any connection to the Cherokee. Lawrence and Kemp extracted mitochondrial DNA from 63 individuals. 55 samples came back usable, a strong result given the age of the material. The haplogroup frequencies themselves were unremarkable. What produced an unexpected result was the principal coordinates analysis. This technique positions populations in a multidimensional space according to how similar their maternal lineage profiles are, with closer positions meaning more shared ancestry. The Arikara did not land near the Pawnee, their closest geographic neighbors. Nor did they cluster near the Mandan, who shared the same river drainage. Their deepest genetic affinities pointed toward the Siouan-speaking nations and the Cherokee. 3. Groups separated by hundreds of miles, belonging to 3 language families with no recognized common ancestor, were genetic neighbors in that deep maternal space. There is a controversial proposal in historical linguistics called the Macro-Siouan hypothesis. It suggests that Iroquoian, Siouan, and Caddoan languages all share a common ancestor too remote to be visible in surface vocabulary or grammar. Most specialists have treated it as speculative. Lawrence and Kemp's clustering result did not prove it, but the genetic data made it considerably harder to set aside. Taken together, the evidence establishes a consistent pattern. Cherokee maternal lineages carry a northern genetic signal concentrated in the Great Lakes corridor, consistent with a population movement into the Appalachians during the late Archaic period. The old assumption of in-place southeastern origins cannot account for the haplogroup X distribution, the Arikara clustering result, or the precision of the overlap between genetic and linguistic geography. But the honest account of this evidence requires acknowledging what it cannot resolve. Mitochondrial DNA traces only one inheritance path, the maternal line, one thread out of the full web of ancestry any individual carries. A community could display a northern maternal signature through sustained intermarriage over generations without undergoing a wholesale population displacement. The difference between a large-scale migration and a slow process of gene flow across centuries is not visible in mitochondrial data alone. Resolving that distinction requires genome-wide analysis, the kind that captures ancestry from every direction simultaneously. For the Cherokee homeland, that data is almost entirely absent. The sampling problem compounds the gap. Most Cherokee genetic studies have drawn on citizens of the Cherokee Nation in Oklahoma, descended from survivors of the 1838 forced removal. Approximately 16,000 Cherokee were marched west to Indian Territory that year. Roughly 4,000 died in transit, 1 in 4 people. A mortality event concentrated into a single season is a severe genetic bottleneck. The profile of Oklahoma Cherokee today reflects the specific individuals who survived that march, not necessarily the full range of lineages present in the pre-contact population. The Eastern Band of Cherokee Indians in North Carolina offers a different vantage point. Their ancestors remained in the mountains, avoiding removal by retreating into terrain the Army could not easily penetrate. Their lineage profile has not been filtered through that bottleneck in the same way. Preliminary comparisons between Eastern Band and Oklahoma Cherokee populations suggest detectable differences in lineage frequencies, which is exactly what bottleneck theory would predict. The Eastern Band community is small, though, and comprehensive sampling requires a collaborative research relationship that takes time to build. That relationship problem runs deeper than logistics. For decades, genetic studies on Native American populations were conducted without the consent of the communities involved. Samples were extracted from burial sites without permission. Findings were published that contradicted oral traditions, and in some cases cited in legal proceedings to challenge land rights and tribal status. The Cherokee Nation now maintains formal protocols requiring genuine research partnership from the earliest stages of project design. Those protocols are not obstacles. They are the framework within which this kind of research can be done legitimately. Several collaborations structured on those terms are reportedly in development. When they produce results, the question will shift. Not whether the Cherokee came from the north, but how. A single sustained migration, arriving over a generation or two. A gradual drift southward along the mountain ridges across centuries. Multiple waves. The genetic record as it currently exists cannot distinguish between those scenarios. The ancient genomes that would make the distinction visible are still in the ground. Somewhere in the river terraces and rock shelters of western North Carolina and eastern Tennessee, beneath forest soil that has not yet been systematically sampled with modern methods, there are remains that may carry the actual genetic signature of the communities who first brought an Iroquoian language into these mountains. The language held the memory for 4,000 years. The DNA is only beginning to catch up. What it will find, when the conditions are finally right to look, is the part of the story that has never been written down.